Hey everybody, how's it going? This is Mr. Gosland here. Unfortunately, we've gone to remote learning or distance learning, so we're not able to have this lesson here in the classroom. But I still wanted to give it for you guys so that you have it to refer to. Today, we are beginning our unit on evolution. We just finished up a unit on classification. It was a short unit, but that's going to tie in perfectly to our evolution unit. So I'm going to do this video with a PowerPoint that we had that we were going to go through in class. And this PowerPoint goes in tandem with this assignment that you're going to be doing that's posted on Canvas for evidence of evolution. I will give you time throughout this recording to go ahead and work on the assignment. There will be moments where I'll tell you can go ahead and pause and work on it, or you can watch the whole thing through and then go and finish the assignment after that. So let me go ahead and share my screen with you so that we can get the PowerPoint up here. Let's see. Oh, I don't know if that worked. Let's try that again. There we go. Okay, so this is the first portion to our evolution unit. We are going to be talking about evidences of evolution. It's kind of like an intro for us. I know that you guys, or you should have learned a little bit about evolution in 7th grade, I believe it was. So we're going to just cover it a little more in depth right now. So the first very important thing to understand and realize before we dive deeper into evolution is theory. There are two types of theory that we are going to talk about. The first of them is regular theory. Regular theory is just going to be our day-to-day use of the word. We use the word theory a lot. I use it a lot when I say things along the lines of, in theory, I think this, or my theory about this situation is this. My theory is not scientific. My theory has no evidence to back it up. It is simply just a personal opinion that I have. So regular theory is a hunch or a guess that we have without any supporting evidence to back that up. Now, on the other hand, we have scientific theory. Scientific theory is very different. And for our unit about evolution and for this course, it's going to be very important to understand these differences. Regular theory does not have evidence. Scientific theory is well supported. There's a lot of evidence to back it up. When we think about evolution or anything scientific in general, we think about the evidence that backs it up. You don't have, when we studied our genetics unit, you don't have all the information about genetics and Mendelian genetics, for example, with the peas. There's not that information there in science without the evidence to support it. So in evolution, it's going to be the same thing. We need well-supported evidence and well-documented. And this evidence is going to be everything in the natural world for us. It can include laws, hypothesis, and facts. Okay, so we are going to watch this video real quick. It's about 5 minutes long. And this is going to talk again about theory versus theory, scientific theory versus our casual or our regular theory. I like this video. I feel like it just explains it better than I can. So we're going to watch that really quick. Let me get it going.
Some words used casually have different meanings than when used scientifically. I'm going to give you an example. When I was younger, I noticed that sometimes after it rained, I'd find earthworms in puddles, and I'd wonder, why are these earthworms coming out of the soil when they're safe there and risking their very lives in these puddles? I mean, I was pretty sure they didn't like being in the puddles. They were getting stuck in them for some reason. Keep in mind that this was before the age of Googling information, and I came up with all kinds of ideas. Maybe the earthworms actually were crazy about water, but they didn't know how bad it was out there until they found themselves stuck in a puddle. Maybe when it rained, the water flooded their underground tunnels, but it somehow confused or disoriented them, so instead of digging down, they dug up, launching themselves into a puddle. Either way, it was my job to rescue them, because, hey, earthworms are pretty cool. Something I can add to my resume. My ideas about why earthworms ventured out when it rained were not really correct. If anything, maybe one of these ideas could have been used to develop a hypothesis. A hypothesis can be defined as a suggested explanation that I could then test. But see, back when I was a young kid, I would have told you that these were my own personal theories. And that's the problem with this word theory. The word theory tends to be casually used in this way in everyday life all the time. I'm sure you've heard your friends say before, I have a theory about that. When using the word theory in everyday life, theory might be an opinion, hunch, or guess. You might say you have a theory about why there's a water stain on a ceiling or why sushi is amazing, but it's important to understand that you are using the word theory very casually. See, a scientific theory is a very different thing. A scientific theory is an explanation supported by scientific evidence. It's fortified by facts. It's been tested repeatedly. And if you want a more detailed definition of a scientific theory, we have a link in our video details to recommend. The thing is, a scientific theory cannot be dismissed as just a theory. In order to reach the level of scientific theory, a scientific theory must be backed by science facts and evidence supporting it with repeated testing. There are many theories in science. There's a theory of the atom, atomic theory. There's the theory of general relativity. There's the cell theory that we frequently talk about in many of our videos. Now, while scientific theories can be disproven or modified, it is important to understand that the word theory in science has such a different meaning than the casual use of the word theory in everyday life. When I was younger, I used to think that theories, one day, could graduate into laws. I think my misconception had something to do with learning how a bill becomes a law, and somehow I thought scientific theories followed a similar path. I'm really not sure where I got this misconception. But in case you have it too, scientific theories do not graduate into scientific laws. They can't, because they're completely different things. One is not more powerful than the other either. The word law might sound more fancy, but it's not. They truly are just different things, but they are both very important in science. Scientific laws tend to describe a natural phenomena, whereas a scientific theory can provide a scientific explanation for it. Many scientific laws are even represented mathematically. For example, Newton's second law of motion shows how acceleration is related to the force and mass of an object. It can be written mathematically here, but as a law it tends to describe, not explain why. Since we tend to specialize more with biology, let's not forget about Mendel's laws. He's often called the father of genetics. He has 3 laws that you can explore: law of segregation of genes, law of independent assortment, and the law of dominance. These laws describe phenomena happening with his pea plant experiments. And while these laws each describe a natural phenomena, they don't give an explanation of why the phenomena happens. It's a side note that actually would have been really hard to do, as DNA wasn't yet understood in his time. Understanding how the casual use of the word theory differs from the scientific use of the word theory, as well as understanding how theories and laws are different things, are all very important in science. Sometimes I wish science had a different word from theory, just because the casual everyday use of that word is so different from the scientific use of that word. And before we go, in case you also have been curious this whole time about why earthworms may come out when it rains and find themselves in puddles, check out some further reading suggestions that mention different researcher hypotheses in the video description. It's pretty fascinating. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. So that taught us a little bit more about the difference between theories. I found it very useful. It's important, like they noted, to understand that along with theories, there are certain theories, like our casual theories, that we can take and turn into a hypothesis if we want to research something. So just like she had the question about the earthworms and why they come up to the surface when it rains, in her mind, that was a theory, but in reality, it's not a scientific theory. She can therefore develop a hypothesis of it and say, I think they come to the surface because of this reason. but that doesn't make it scientific until she does that research and does that testing and studying to have evidence to back it up. Once you have that evidence and you can say, we have this evidence and show this, at that point, it could become a scientific theory. Doesn't mean it will, but it could once you go through the testing. So we are going to talk next about Charles Darwin and his theory. Charles Darwin is a very famous name. I'm sure all of you have heard of him. He kind of brought about this whole race of evolution and let's study it and figure out what it is. So we're going to talk about his theory and exactly what he thinks. So that's him right there. He's got a lovely beard going on. Charles Darwin's theory of evolution has three main components to it. The first of which is that variation occurred randomly among members of species. With that being said, evolution is not something that is scheduled or planned. We don't plan to have evolution. We don't say, I've got this group of species over here, and now I want them to transition into either this group of species, or I want them to make this change to make them better. That's not how it works. Everything in evolution pretty much is random. His second component to evolution is that individuals' traits could be inherited by its progeny, meaning you've got mom and dad, they have offspring. This is how those traits are passed along, is that we pass down those traits. Just like we studied in our genetics unit, when we look at pedigrees and when we do Punnett squares for X-linked or sex-linked traits or alleles, we can look at how those traits are passed down. Let's say that I have brown eyes, my wife has green eyes, and we have 3 kids. What's the rate that they'll have brown or green eyes? Or will those go away altogether over generations and generations? That's what Darwin was getting at. His third component is that the struggle for existence would allow only those with favorable traits to survive. We'll talk about this more in kind of our next section that we're going into in a week or so when we talk about natural selection. But this is basically saying that species evolve so that they can develop, or they evolve with those favorable traits to help them survive. So if you've got one animal predator-prey situation, you're going to have a prey that develops an ability to either camouflage or to run faster. And then you're going to have a predator who also develops things like camouflage or speed because they need to keep up with each other. He needs to develop that to catch his prey. The prey needs to develop to get away and survive. So that's just how it's looked at, is that the favorable traits will be passed on. Darwin also thought of evolution as a descent with modification. So when we talk about family or pedigrees, you've got parents, offspring, 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 and that's kind of our descent through the generations. Evolution, as Darwin thought, had modifications to that. Descent with modification, his definition is a process in which species change and give rise to new species over many generations. Evolution is not something that happens quickly. We know this. It takes a long time for evolution to happen. And we'll talk about more specific occurrences of this. But if we had, let's see if I can do this without my head getting chopped off by the video. So as far as descent with modification goes, if we had, this is one organism, a circle. Over generations, we may see a slight change, and now they've got a little indent. This is a terrible example, but I'm using it to show the change that could occur. And over a couple more generations, we may see that this changes a little bit more. And a few more generations, we see that this could change a little bit more. Now, this isn't a great example just because there's really no reason why it would change, no favorable trait there. But that's just what it means, is that over generations, we can see species change, and that's where the descent with modification, the indent here being that modification, and the linear line right there being the descent among the generations. All right, let's move on. Okay, so this was a part in class where we were going to discuss it. I want you to think about, why is Darwin's theory a scientific theory and not a casual theory? You obviously don't have a partner to share with, so just think about it to yourself for a minute. Maybe jot a few notes down that you might think of, or a few questions, and we can talk about them when we do get back in class. But think about it for a second. Why is Darwin's theory a scientific theory and not a casual theory? Okay, I know that wasn't the full 30 seconds. You can pause the video and keep thinking or writing notes or ideas down if you need to, but just kind of keep that in mind as we go through it. Why is Darwin's theory scientific, not casual? So we are now going to get into more of what is evolution, kind of an intro to evolution. Now that we understand the theory behind it, we want to look at what it is. So evolution is a change in the genetic makeup over the genetic makeup of a population over time. So just like we saw with my example right here, over time, we saw the change in traits. That's evolution. There are different types of evolution: macroevolution, which is large scale, and this occurs over a very extended period of time to form new species or groups. Or we also have microevolution, which is more of a small scale and affects maybe one or a few genes. And this happens in populations over a shorter time. So we're going to look macroevolution first. An example of macroevolution would be something we look at like fossil records, which we'll talk about in a little bit. Macroevolution or fossil records, we can see how, for example, horses have changed over time to either develop into new species or new groups or breeds of horses. This is not something that happened in a year or a decade. This is something that happened over millions of years. Microevolution could be something that we could observe on a closer scale. One really good example of microevolution is drug-resistant bacteria. So when we have bacteria, for example, when you get sick, let's say with strep throat, and you go to the doctor and they prescribe you with an antibiotic, that antibiotic is supposed to help you heal, because that's what kills the bacteria. However, we have some bacteria that has learned to evolve quite rapidly and change their genetic makeup to not be affected by those antibiotics. Another great example of this is COVID. We're going through it right now with our vaccines. We see and hear all of these variants: Omicron, Delta, just the regular COVID that was discovered first. And we developed vaccines to protect us from those or from COVID. But as time has gone on, we've seen the variants appear, and that is that virus making changes to their DNA and evolving in this short period of time, or going through microevolution, we could say, so that it can survive. So that's one example that's pretty relevant to us now. We are going to discuss some misconceptions about evolution. There are a lot of misconceptions about evolution, and we are not going to cover all of them in this lesson. So I would invite you to please write down your questions so that when we return to class, we can discuss them. If you want to, you're more than welcome to email them to either myself or Mr. Gisman, and we can look over them and then kind of incorporate your questions into our next lesson. So let's see. Misconception number one is that it does not produce perfection. Just because something is changing, like we see back here, does not mean that going from here to here is a perfect change. It may be a favorable trait and it may have helped, but that does not mean it's perfect. We will still see problems or errors occur. And we see that with all evolution, with all species. It's never a perfect scenario. Let's see. Oh yeah, that's a good point to cover. I don't want to skip by that one. Evolution and natural selection is simply to get those genes passed into the next generation. So it doesn't mean we're passing on the perfect genes, but we need to pass those genes down because we want to create offspring. That's our goal of survival, is to have offspring that will survive. So we may not be perfect and we may not have those favorable traits to survive, but as long as we have offspring, our lineage, our line, or our descent is going to survive. The next misconception I want to talk about is that natural selection or evolution is not a random process. It's not perfect, but it's not random either. And the reason I say it's not random is because as a species or as organisms, we are looking for those ways to survive, meaning our genetics and our bodies are evolving to find those ways. So just like we talked about with predator and prey, we're looking for those ways that we can survive, those favorable traits. That means that it's not just random, like, oh, all of a sudden we're faster anymore. That's not how it works. Our body is working up to being faster. It's not just all of a sudden we're faster in the blink of an eye. Our body works towards that. We gain endurance so that we can have that. And then as we have offspring, and more generations down the line, eventually they will evolve and be faster and faster and faster. So it's not random. There's a reason to it. Let's see. Yeah, so just like I said, it aids in survival. Natural selection is a process, not a guiding hand. So it's not like we're going to look at one species and say, we want this species to evolve and have these traits. That's not how it works. We don't guide how we want things to evolve. It's just a process that happens naturally, hence the name natural selection. This is a really good graphic. Variation plus differential reproduction, or randomly, plus heredity equals the natural selection. So you need a variation in genes, you need that random reproduction, and you need the heredity or the passing on of those genes in order to have natural selection. In this graphic, the polar bear says that he needs a good fur coat, and that's not how it works. Just because he wants it doesn't mean he'll get it. They developed over time to resist the cold and to develop those things that they needed. So how can we observe evolution in species? We are going to talk about, I believe there's 5, anatomy, molecular biology, biogeography, fossil record, and direct observation. Not necessarily in that order, but that's what we're going to talk about. So first, we're going to start with anatomy. We can use anatomy. Actually, it's really important to use anatomy with evolution. Sorry about that. They're making announcements over the intercom. We can use anatomy to trace organisms back to their common ancestors. So if we look at this right here, and this is what's called a cladogram. If you look towards the bottom, you'll see at the very bottom of it, A, B, C, D, E, F, and so on. That would be a common ancestor. That would be one organism or one species that existed, and all the branches going up off of it is how species evolved. So let's take, for example, the letter A. You've got down by A, the common ancestor, and if you follow up the line that goes to the left, all of those little marks along the line are different organisms that have been evolving. And then all of a sudden you get to where it breaks off. Well, that's where a big change in evolution has occurred. And we've all of a sudden got 2 new species happening. And you follow both those lines up and then they break off. You've got one that goes 3 and one that goes 2. Sorry, I might hold my hands too high. And those are new species. So when we talk about having a common ancestor, or how Darwin suggested that we all came from apes, that's not necessarily what we're saying. we are saying that we may share similar anatomy or similar genetics with a common ancestor, and this is how we draw that out. So like it says, we use this idea to work backwards and see how we can be related. And we'll go into some detail on what types of things we look for when we're looking for those common ancestors. So the next part of anatomy is homologous features. So we know the word homo means same. We learned that in our genetics unit. And the word logos means info or information. So when we say homologous, we're looking at the same information. So the same features, shared physical features that could link a common ancestor, just like in this slide. We're going to look for the same features that letter A had down at the bottom, that maybe the very top row now has. So this is something we can look at. This is a fairly common graph that we look at, or illustration, I should say, for homologous features. We have a human bone structure, a dog, a bird, and a whale. Now, if you were to spend some time studying these, you would notice a lot of similarities, and they've done a really good job at color coding this for us so that we can see where those similarities are. You'll notice down where, for example, the humans have our fingers is colored dark brown. All the other pictures that are colored dark brown in that same area, those are the same types of bones and skeletal structures. The yellow above that, the same, the red and the white and the brown above that, or the tan. Those are all the same bone structures, but they've evolved. For example, if you look at the human and the whale, so on the far left and the far right sides, they look very, very similar. And that's how homologous structures or homologous features have evolved over time, and how we can trace things back to common ancestors by saying, we all have this. Now, that's not to say all organisms have these same structures, but to look at it and say, these 4 organisms have it, they must have some common ancestor in the past. Let's see if I can switch slides. Okay, so at this point, we've talked about some anatomy. We've talked about homologous features with anatomy, I should say. In your worksheet or your packet, the very first portion says Part A, homologous features. We are going to work on that. I'm not going to do it with you on the slide. If you want to, you are more than welcome to pause the video and work on and finish Part A, and then press play and continue on. Or you can finish the whole lecture, the whole video, and then go back and do your packet. So just to give you a little brief overview, Part A talks about homologous structures and talks about their meaning and what they're used for. And it's going to ask you to examine these homologous structures, kind of what we just looked at on the slide, and to fill out this chart about their different functions. And once you do that, there are 4 questions on the 3rd page that it's going to want you to answer regarding homologous structures. Wants to know the functions, if they're arranged in a similar or different way, what does the similarity suggest, and talking about possible explanations of this. So if you want to pause the video, you are more than welcome to, and you can go ahead and work on Part A and then continue. I'm going to move on if you would like to follow with that. Let's see if I can get past this video. Sorry, I didn't change my PowerPoint from the in-person lesson to this. So the next portion we're going to talk about is that not all physical features are a sign of common ancestry, such as homologous structures are. So we know homologous structures point to common ancestry, but there are other structures such as analogous features. And analogous features evolved independently in different organisms because the organisms lived in similar environments or experienced similar pressures. So for example, if we were looking at predator-prey again, they both may have evolved to be super fast. Like let's look at the ostrich and the cheetah. They are both super fast animals, but that doesn't necessarily mean that they share a common ancestor. Now, I don't know that for sure if they do or not. I haven't looked at that. But just as an example, just because they're both fast doesn't mean that they share a common ancestor. They both may have experienced selective pressures that were the same from where they were living, and that could have been the reason that they evolved to do that. But that doesn't mean that they share a common ancestor. So another good example of analogous features would be the Arctic fox and the ptarmigan. It's a type of Arctic bird, and I don't know how to say it, so I apologize. They both evolved with the ability to color change in this different seasons. So both the Arctic fox and this bird have the ability to change their either feathers or their fur coat to be a bright color of white during the winter seasons when there is snow on the ground. However, that does not mean that they are share a common ancestor. You're not going to see this bird and this fox share a common ancestor like that just because they both color change. Their ability to color change is not a good distinction of a common ancestor as a homologous structure like bone feature would be. So this is a picture of them with the white feathers and the white fur. So that is analogous features. Again, just like we did with part A, if you'd like to, you may pause the video and you can complete part B, analogous structures, in your packet. This one is going to have you comparing a butterfly wing and a bird wing to look at their different bone structures, and it wants you to talk about what their function is of the wings and what the wing functions share, what ways do they differ, and explain why they are considered analogous structures and not homologous. So just 3 little questions to answer on that one. It's not too difficult. So you can pause it and do that now if you'd like, or you can move on. Go. Okay. So we've talked about homologous, we've talked about analogous, and now we are going to talk about something called vestigial structures. So we have organisms that have homologous structures with a different name known as vestigial structures. And vestigial structures are different features or structures on an animal or an organism that have lost their function, their previous function, or the reason that they were there originally. So you look at certain things that may have lost the ability to walk, so they don't need their legs anymore. A vestigial structure would be a remnant of that leg. And we'll look at that in a little bit. Oh, right here. So some examples would be tailbones of humans. We don't use those. We don't have a tail, but there's evidence based on our skeletal system, based on our homologous structures in our skeletal system, that shows that we had some use for it in the past and it's just left there, type of thing. Hind leg bones in whales. Whales obviously swim in the ocean. They don't walk on land. They lost that use for walking and have kind of, for lack of a better term, absorbed those legs and don't need them anymore. But there is evidence that they were there based on their skeletal structure. And another really good one that we'll look at a picture here is underdeveloped legs in snakes. So this picture right here, you see that they have vestigial legs of a boa constrictor. There's 2 tiny little legs in the picture. So snakes used to walk, some of them, and they don't need to anymore. So over time, they have evolved out of that ability. In other words, the legs have disappeared in a sense, and they don't use them. So that would be vestigial structures, structures that are not useful to us anymore. So same thing with A and B. There's part C, vestigial structures, in your packet. This one is going to talk about something really cool, and maybe we'll see a video of this in a later class. It's going to talk about cave fish that have lost their ability to see. They are blind, and it wants you to talk about why that may have happened. So it's got 2 questions to talk about why eyesight was not important to the cave fish, and to talk about common ancestry based on that. Now, it also has a chart in here. And this one is not about the cavefish. This chart is about vestigial structures in humans. So things like our appendix, our tailbone, our wisdom teeth. And it wants you to talk about probable functions of those structures and why they've gone vestigial. Why don't we use them anymore? And then apart from part C, there is a section called overview questions. And there are 3 questions on there that you can go ahead and answer. So go ahead and pause your video and work on part C and on your overview questions, and then you can come back and we'll keep going. Okay, so the next thing that we are going to discuss is called comparative embryology. So comparative embryology has to do with our embryos and our embryo structure, the way we develop as like a fetus. So comparative embryology has become over time very important for us to study because it allows us to look at organisms as they are developing. And there is a lot of organisms as we look at their embryos and their development that help us to look and see that common ancestry between them and to see that evolution happening. So like it says here, some homologous structures can only be seen in the embryos. An example would be the vertebrate embryos. Humans, as we are developing as a fetus in the womb, we have gill slits that go from our ears to our jawbones is kind of where they develop eventually and disappear. And those gill slits and also our tails or our tailbones develop, but then disappear by the time we're born. So this is something interesting to look at because it shows common ancestry to other organisms. Other organisms such as fish, reptiles, and birds also develop gill slits and tails as they're developing. So comparative embryology has become a really important part of evolution and studying that. So that was a little short section. I want you to go ahead and do part D of your packet. That's in Canvas. You are going to be studying this chart in your packet. And this chart has a human, a monkey, a pig, a chicken, and a salamander. And it shows 6 stages of their embryonic development. So you will study and observe that chart. And then there are 5 questions relating to part D for the comparative embryology. Want you to list some differences, want you to compare and contrast different ones. Let's see, and it wants you to talk about and make some conclusions about different evolutionary relationships based on what you see in the comparative embryology or in the embryo development. Okay. We are going to talk about biogeography really quick. Biogeography, this part is not in your packet, but it's really interesting to look at. And part of it has to do with Darwin as well. So biogeography would be our geographic location or the geographic distribution of organisms. When you look at organisms that have been distributed all over the world and then maybe segregated in that area of the world, kind of confined in there, you can look at their shared similarities based on their geographic location. Some of them, like for example, if we look at Australia, we used to have Pangea, one big continent, and as it broke off, Australia ended up over here, isolated from the rest of the world. And it's interesting to look at the mammal species in Australia because most of them are marsupials, which means that they carry their young in the pouch as opposed to nourishing them through a placenta in the womb. And it's interesting to see that off in this continent or this country that most of them are the same type of animal, marsupials, as opposed to the rest of the world. And you have to think and look at the evidence of what type of evolution occurred here to make them have those changes and to all be similar. So there's some different ones like the koala and the kangaroo. We also can look at the Galapagos Islands, where Darwin studied some animals. He studied finches and tortoises. One of my favorite ones to look at is the finches. Galapagos Islands are kind of isolated again, like Australia was or is. And It's interesting to look from island to island based on the food source that was available to these finches, how over time their beaks have evolved and changed. Some of them have soft berries that they can eat. Others have hard nuts. Others have different types of foods that require a different beak for them. You'll notice the one in the top left has a very strong looking beak. He's probably a finch from the island that only has nuts for them to eat, and he needs that power of his beak to crush them. And they've evolved over time to do that. So that is biogeography, studying how isolated species evolve together versus the rest of the world. We're going to talk about fossil record. This will be part E of your packet. We can look at fossil record in different scenarios like this, where you have the different layers of the earth. Fossil record is not complete. We may have found a lot of fossils, but we have not found near the amount that there are out there. It's also hard because fossils are very fragile. So when you study a fossil, you can only get so much information out of it based on the condition that it's in. The older the fossil, the harder it's going to be to find and the worse condition it's in. Along with that, most of the organisms that have lived on Earth never fossilize. The conditions are not right. You need perfect conditions to be able to fossilize. And most of the ones that do, we just never find. So there are different ways that we can determine how old fossils are that can help us study. First would be the rock layers called the strata. In this image on the screen, you see strata in there, and each of the lines shows a different age of that fossil. So you've got 2 million years ago, 100 million, 400 million, 500 million. Each of those strata are different ages of when that organism died. And that can help us studying the strata and the rock layers to determine how old fossils are. The new layers are on the top and the old on the bottom. We can also use something called radiometric dating. And this is how you measure the radioactive decay of a fossil. And it can tell you how old that fossil may be. Now, it's not going to be perfectly accurate. It's not going to give you the exact number or age of a fossil, but it gets us close so that we can look at it. And the reason this is helpful is to help us study evolution over time as we study similar fossils of animals. So for example, this one, equestrian, equus, horses, we can study down at the bottom. You'll see Mesohippus all the way up to the top where it's equus. And that's how horses have evolved. We can study based on the strata that they're found in or the radioactive dating of them. You can see how they have evolved over time. They've gone from something really little to something fairly large. You can also look on the right-hand side of the image, the bottom. Horses used to have 3 toes, and they evolved into just having 1 large hoof. Now, why that was necessary to evolve that way, we may not understand, but it's interesting to look at that progression as we find those fossils. And that's why fossil records can be so important for us to study. So part E of your packet right here is going to talk about the fossil record. It has kind of a little history that you can read about fossil record and absolute dating and how they date it. It's also got the same image that was on your slide, studying the strata and the layers of the rock. And it will have, let's see, I can just tell you here, 6 questions about fossils. So it wants you to study and observe the diagram, talk about how you know which fossil's older or newer. And then it has some kind of questions in there that get you thinking a little bit about why some fossils are the way they are when we look at their evolution from then until now. So go ahead and pause the video if you want, or do it after. Okay. And this, I believe, is the last one that we were talking about. And this is molecular biology, or molecular evidence, I should say. That would be a better way to say it. So just like being able to study homologous structures or analogous features and stuff, we can look at evolutionary relationships through their genetics. Just like we studied in our genetics unit with pedigrees, we can look at genetics to help us determine how organisms have changed over time. We can look at their DNA, their same genetic material. We can look at similar genetic codes that they may have and gene expression that may be turned on or off as we study it. And this is something that's been interesting because it's not something that's very well known how to do. It's something that's still being developed that we're looking into on how to read the genome. You can look at their amino acids and their building blocks to help you learn more about them. And this can be very important in studying common ancestry, just so that we know, hey, they share this organism and this organism shared this portion of their genome that helps them do these things. So it's something we haven't had in the past that's growing exponentially now. So in order to practice that, you will have the opportunity in your packet with part F to look at some different organisms. And it looks like you're going to be looking at a whale, a sperm whale, a porpoise, which is like a dolphin, a giraffe, and a hippo. And you are going to be studying a portion of their genome like that. And you are going to be looking for the differences between them. I just want to explain this chart to you a little bit. It says, determine how many nitrogen bases are different between each pair of animals and complete the chart. So when you look at this chart, you're going to look at it across and down. So for example, you're going to look at the sperm whale right here compared to the regular whale and put how many differences you count in that box. For example, right here, the hippo compared to the porpoise, and in this box, write down how many differences between the two genomes you found. And remember, when it talks about nitrogen bases, we're looking at those pairs or segments of 3 in their genome. The 3 is a nitrogen base. So you're going to want to pair those off to look at them and fill that out. Now, I know that that was quite a lengthy video to watch, and kudos if you got through that. I really wished that we had the chance to observe or to go over this lesson in class with you. I wanted to give you the time to work on it in class since it is kind of a large packet to get through. But I want everyone to go ahead and do their packet and submit it through Canvas so that we can take a look at this. And you'll want to do this packet because it's gonna play a particular role in our next project that we're doing with some different evidence research and stuff. So if you have any questions, feel free to email me, agosland10@graniteschools.org. And if not, I guess we'll see you in class when we all get to meet together again. Go ahead and finish the packet. And thank you for watching the video. And let me try and get this unshared now and turned off for you. Let's see here. Okay, thanks guys. We'll see you soon.